This lecture video is on chapter 15, which is the second part of glycogen metabolism. So before Thanksgiving, I provided you chapter 15 notes. What you want to do is go to those notes and go to slide 15 of those notes, which covers glycogen metabolism. So we will finish glucose regulation in class, and the lecture video is on glycogen metabolism. So glucose, I like to think about it as ATP cache. Glycogen is like an ATP savings account because this is how glucose is stored and it can be quickly released to get energy from it. Glycogen is stored either in the muscle or the liver tissues. Glycogen in muscle is a glucose reserve for exercise. It's released in response to epinephrine, and it becomes metabolized through glycolysis once the glucose residues are released. Glycogen in liver has a different function. When it's released from the liver, the glucose is important for maintaining blood glucose levels. It's released in response to the hormone glucagon, and this released glucose is going to be metabolized by tissues other than the liver. So, for example, the brain is going to be a major utilizer of glucose. Remember that glycogen has a highly branched structure that allows fast access to glucose. Glucose units can only be released at the non-reducing end, that is the terminal end of a polysaccharide chain. Glycogenolysis is the process of breaking down of glycogen, so that's where the terms glycogen and lysis come from. It's the process of converting glycogen to the molecule glucose 6-phosphate. Glycogenolysis occurs in response to low blood sugar or during exercise. Glycogenolysis is stimulated by hormones epinephrine or glucagon. The enzyme that catalyzes the first step of glycogenolysis is glycogen phosphorylase. This enzyme breaks the alpha-1,4 glycosidic bond between the two glucose residues at the non-reducing ends by attack of an inorganic phosphate. When there is bond cleavage by addition of an inorganic phosphate, this is called a phosphorylasis reaction. And so this is distinct from hydrolysis, where a molecule of water is added to break a bond. In the reaction, inorganic phosphate attacks carbon 1 of the glucose residue that is at the non-reducing end. This causes the rest of the glycogen polymer to leave, resulting in glucose 1-phosphate and then glycogen that has the remaining number of glucose units. So it's going to be N-1. This step that's catalyzed by glycogen phosphorylase, which is breaking the alpha-1,4 glycosidic bond, is the regulated step in glycogenolysis. And just a note that glucose residues that are at alpha-1,4,6 branch points are removed by debranching enzyme. Once we have glucose 1-phosphate is converted to glucose 6-phosphate. And so the enzyme that catalyzes this reaction is phosphoglucomutase. And this is done by an isomerization reaction where the phosphate attached to carbon number 1 in glucose 1-phosphate is transferred to carbon number 6 to make glucose 6-phosphate. This is a reversible reaction, and so just a note that this same enzyme is actually used in glycogen synthesis. Once we have glucose 6-phosphate, it can have two different fates depending on whether glycogenolysis is occurring in the muscle or the liver. If glycogenolysis is occurring in the liver, glucose 6-phosphate is going to go through a hydrolysis reaction that's catalyzed by glucose 6-phosphatase. So this is the same enzyme and reaction that we've already seen in gluconeogenesis, where glucose 6-phosphatase is going to hydrolyze the phosphate off glucose 6-phosphate to make glucose. This glucose is then going to be exported from the liver into the blood circulation to then maintain blood glucose levels. Glucose 6-phosphate in the muscle has a different fate. Instead, glucose 6-phosphate actually goes directly into glycolysis in the muscle, where the muscle will use the released glucose 6-phosphate for generating ATP through glycolysis. Glycogen synthesis is the process of converting glucose 6-phosphate to glycogen. It's not exactly the reverse of glycogenolysis because it uses almost an entirely different set of enzymes and reactions, except for one. Glycogen synthesis occurs during the fed state in response to high glucose levels. Glycogen synthesis occurs primarily in the liver and the muscle tissues and is stimulated by insulin. Glycogen synthesis also requires hydrolysis of a nucleotide triphosphate, specifically uridine triphosphate. The first step of glycogen synthesis is the conversion of glucose 6-phosphate to glucose 1-phosphate. Just to take a step back, most exogenously derived glucose is faded for glycolysis. So remember that in the first step of glycolysis, glucose is converted to glucose 6-phosphate, which is catalyzed by hexokinase. This glucose 6-phosphate can then enter into glycolysis to generate ATP. However, in the fed state, there's an excess of glucose. So some of the glucose is made into glycogen, where glucose is converted to glucose 6-phosphate, and glucose 6-phosphate is converted to glucose 1-phosphate, which then, through several additional steps, can be converted to glycogen. The enzyme that converts glucose 6-phosphate to glucose 1-phosphate is phosphoglucomutase. Once glucose 1-phosphate is formed, it is then activated to make UDP glucose. In this reaction, glucose 1-phosphate attacks the alpha phosphate of uridine triphosphate, resulting in pyrophosphate and UDP glucose. This reaction is driven forward because pyrophosphate further hydrolyzes to inorganic phosphate, which is a thermodynamically favorable reaction. You might ask, why is UDP glucose being made in glycogen synthesis? This is because formation of UDP glucose is metabolically irreversible. This is due to the fact that pyrophosphate hydrolyzes to inorganic phosphate, which makes a reaction irreversible. The nucleotide UDP is a good leaving group in the reaction, which is very important for the next step. And also, by taking the sugar with nucleotide group, the cell knows that the sugar is destined for glycogen synthesis. So this is able to separate the glucose pools for utilizing it for different purposes. Once UDP glucose is formed, the glucose residue is then incorporated into glycogen by glycogen synthase. In this reaction, glycogen synthase transfers the glucose residue from UDP glucose into the non-reducing end of an existing glycogen chain. So in this reaction, the hydroxyl group that is attached to carbon number 4 of a non-reducing glucose residue in glycogen attacks the anomeric carbon of UDP glucose, resulting the release of UDP and incorporation of the glucose residue into the non-reducing end. This makes a new alpha-1 4 glycosidic linkage. The step that's catalyzed by glycogen synthase is the regulated step of glycogen synthesis. So how can we ensure that both glycogen synthesis and glycogenolysis are not running at the same time? The way that glycogen metabolism can be coordinately regulated is through covalent modification of the enzymes through phosphorylation of serine residues. So both glycogen synthase and glycogen phosphorylase, which catalyzes the regulated step of glycogen synthesis and glycogenolysis, are both phosphorylated, but phosphorylated has different effects on the enzyme. For glycogen synthase, which is involved in glycogen synthesis, phosphorylation inactivates the enzyme, whereas phosphorylation activates glycogen phosphorylase, which is involved in glycogenolysis. Epinephrine and glucagon stimulates phosphorylation and activation of glycogen phosphorylase. When epinephrine and glucagon bind to their cognate receptors, this causes a cascade of events, which we've already learned results in the formation of cyclic AMP, which then activates protein kinase A. Protein kinase A phosphorylates and activates a different protein kinase called phosphorylase kinase, and then phosphorylase kinase phosphorylates and activates glycogen phosphorylase. So phosphorylation of glycogen phosphorylase on serine residues activates the enzyme. When glycogen phosphorylase is phosphorylated, another name for it is called phosphorylase A. And dephosphorylation of glycogen phosphorylase results in the inactivation of the enzyme. So the inactive dephosphorylated form is also called phosphorylase B. In the fasted state or in response to stress, phosphorylation of glycogen phosphorylase promotes glycogenolysis. So this is going to then result in the release of glucose as a fuel source in the muscle and the liver. Glycogen synthase, on the other hand, is oppositely regulated by phosphorylation. When glycogen synthase is phosphorylated, it's less active. So during the fasting state, glycogen synthase is phosphorylated, which then helps to downregulate glycogen synthesis, which then upregulates glycogenolysis. And insulin promotes the dephosphorylation and activation of glycogen synthase, which promotes glycogen synthesis. Just a word on glycogen loading. Glycogen is the primary source for muscle that was at rest and then starts working vigorously. The energy of ATP hydrolysis that's derived from glycogen breakdown is initially produced by anaerobic glycolysis, with the lactic acid product being processed back to the liver as glucose through the Cori cycle. As an athlete becomes well conditioned, the muscle cells actually generate more mitochondria, allowing for more aerobic metabolism of fats and carbohydrates for energy. Switching to aerobic metabolism takes a few minutes, which is why athletes must warm up before an event. So it's estimated that it takes 8 to 12 minutes for an athlete to use up all of the glycogen in the skeletal muscle, although this range varies depending on the intensity levels. Glycogen loading may allow a person to store more glycogen, which they may then be able to carry out anaerobic metabolism for a longer period of time, either at the end of a distance event or for the entire event if the effort level is high enough. However, some methods are dangerously stressful to the heart.